Welcome everyone to this lecture, which is part of the Ways of Knowing class. My name is Sue Sorensen and I'm a professor of English and one of the 7 Ways of Knowing instructors this year. As many of you know, or most of you know, there are several layers to the experimental, fairly radical class, which is Ways of Knowing. The classroom portion, the round tables, which are larger, and the events that involve everybody. First our Bear Clan walk last month, now this lecture. Next week, our film nights, which involve everyone, and which everyone is invited to, and I'll tell you a little bit about those film nights in a moment. I'm honored and humbled to be learning and living here on land that is not mine, that was cared for over uncountable years by the first peoples of this place. I'm grateful for their stewardship of this land. In a moment, I will introduce you to the 2 scientists who will lead us through today's intriguing discussion called Bodies as Neighborhoods, Horrible Happenings and Happy Harmony. A special lecture offered to us by Dr. Rachel Krause and Dr. John Brubacher. But some housekeeping first. Here is the invitation for everyone to join us next week at 1 or 2 or both of the Ways of Knowing films. Next Thursday in the Great Hall, 7 p.m. Last year's film, Flow, winner of the Oscar for Best Animated Feature, which we thought was a good illustration of neighbors, animals as neighbors. And then on Friday at 7 p.m. in the Great Hall of Gods and Men, a very moving and all too relevant French film from 2010, with subtitles, of course, that has won many awards. The true story of an order of monks who live in the midst of a Muslim community in Algeria. And during the civil war, the monks must decide whether to stay with their Muslim neighbors or flee. They decide to stay. So, Ways of Knowing students, you can go to 1 or both of those films. Everyone else is also invited. There might be popcorn. And remember, Ways of Knowing students, you will write a short film review of one of those films that is due in mid-November. And also, Ways of Knowing students, you may wish to take notes today because here's your reminder that you will submit to your professor next week a short response about this lecture. And one last bit of housekeeping. Once Rachel and John are finished with their presentation today, there will be an opportunity for questions, so think about that and be ready with those. So the introduction. I distinctly recall, since I have been here at CMU for more than 20 years, going to the job talks given by these 2 when they were being considered as faculty and thinking, we get to hire these people? God really does love CMU. More important than these 2 being excellent scientists, they are terrific human beings. Each has a wide range of interests. They have a passion for science education that lights up a classroom, a lab, or even an outdoor learning space. They care. They are funny, collaborative, all-round tip-top. Rachel Krause has been at CMU since 2015. She's from BC, but we won't hold that against her. Her specializations are ecology, evolution, and global health. And if you want to talk about fish disease, salmon habitat, parasites, anything at all about the country of Panama, you talk to Rachel. If you have taken a course with Rachel, you know that she wants students from the first year to consider themselves researchers. And she's very eager to work alongside student researchers as a mentor. Rachel is one of the friendliest people I know. She loves to laugh, which is a good thing at CMU, this crazy little university where we try to do big, unusual things that perhaps even big, fancy universities can't figure out how to do. She is that very excellent thing, a good listener. She has been instrumental in this course most of us today are part of, Ways of Knowing. She was one of its first instructors and is now overseeing research, studying the course itself, its methods, and its successes. John Brubacher has taught biology here at CMU for 17 years, but not long enough. He's from Kitchener-Waterloo, but I have never held that against him so far. His His specialty is developmental biology and cell biology, and I have listened to him enough over the years that I know that the tiny, tiny creature at the center of his research is the planarian worm, miracle worms that can regrow missing body parts. I love planarian worms, but John loves them more, although he does slice them up for the greater good. He has published a lot of important research. One of my personal favorites is "A Planarian Nidovirus Explains the Limits of RNA Genome Size," although I have found more useful in my everyday life his paper "Processing Schmittia Mediterranea for Transmission Electron Microscopy, Classical and Microwave Techniques." Microwave, I know what that means. I have not yet read his publication called "A Sex-Specific Transcription Factor Controls Male Identity in a Simultaneous Hermaphrodite," but I'm looking forward to it because it sounds very saucy. Neighbors, please join me in welcoming Rachel Krause and John Brubacher.
Why did I bring a pen up with me? Maybe I'm going to take notes on, well, I can take notes on Rachel's stuff. I'm John. This is Rachel. Sue, now we can all just go home. All right. Welcome to your neighborhood. When we think about our own bodies and selves, we tend to consider ourselves as individual living things. That's what we see, and that's what we consciously experience. And it's not wrong, exactly, but it's not quite right either. Living all over and within your body are trillions of microorganisms. Or just for the rest of the day, to save a couple of syllables, we'll just call them microbes. Here's an example. This is an image of a couple of my cheek cells, but you can also see showing some bacteria on the surface. I counted 3 of them there. Bacteria, as you can see, are tiny, much smaller than a typical human body cell. And if we zoom in closer, here's a photo of bacteria on a grain of sand with some of the slime that helps them stick there. There's still really not a lot to see. Bacteria are single-celled organisms with a very simple anatomy. Most look more or less like jelly beans or gumballs. However, their biochemical diversity, the molecules that they can use as food, and the various designer molecules that they can make from those food molecules are stunning. You are probably home to at least 1,000 different kinds of bacteria. The individual bacteria might say that you're home to, each one of them as a kind. I don't know. Their diverse ways of living reflect their impressive genetic diversity. If you were to count up the number of distinct genes in the community of organisms that lives on and includes you, well over half of those genes would be bacterial. The actual number is probably over 90%. Of the trillions of microbes living in you or on you, your microbiome, most are bacteria, but there are others, and we'll meet some of them today. Various protists and fungi, cells like bacteria called archaeans, and even small animals. Some roundworms and skin mites especially. And for now, at least, I'm going to just save you the description of the little critters that are currently lounging in the roots of your eyelashes. But I'll encourage you to think about them. So, everyone's body in this room is really a living community, a neighborhood. If you can call a population of about 30 trillion microbes plus 1 human a neighborhood. But what are the microbes living in us and on us, if not our neighbors? They are distinct from us, but live in association with us. They latched onto you from the moment of your birth, coming from mom as you left her body, and from the rest of your messy interactions with our living world. Like neighbors that get all caught up in our business, the members of the microbiome are in you and on you, but they are not you. They are not permanent occupants of your body. You shed them with skin cells in spit, urine, and especially feces, which is about 50% bacteria by volume. Every day, some leave, some die, others are born, and still others arrive from various places. The community makeup changes over time, reflecting and perhaps affecting changes in our own habits and circumstances. So, your microbiome and its members are not you, and you are not them. But just like human neighbors, we affect each other, and in fact, we depend on each other in some very significant ways. Going back to bacteria, here's another image of a bacterium. This is a really thin slice right through it, so that we can see some of its internal structure, but again, there's not much anatomical complexity here. But, although these are simple, single-cell organisms, these little critters do interact with each other and with us. They interact, they manage their social lives by taking in and making and secreting a huge variety of molecules. And in that sense, they're kind of like us. Think about the quantity of chemical signals that you emit each day as you breathe, sweat, excrete, shed, spit, sneeze, etc. Now, when it comes to more familiar macroscopic neighbors in our lives, we have some degree of choice about who our neighbors are, or at least which ones we pay more attention to or spend more time with. And that's also true of our macroscopic non-human neighbors. I'm guessing that most of you, if you plant stuff, you plant flowers, fruits, and vegetables in your garden, and not poison ivy. And the animals that we keep as pets are really a pretty small selection of the options that are available to us out there in the world. For a long time, we humans didn't know much about the microscopic neighbors that live on our bodies, or even that they existed at all. Now we do. And as we get to know them, we find that some of them are more likable than others, at least at first. So, there's a temptation to try to optimize our relationships with them, to pick and choose our microbial neighbors. Feeling a little sick? Try this probiotic. You worried about your mental health, your energy, your weight, your memory, your alertness? Somewhere, there is a startup business that wants to sell you a cocktail of microbes for that. But here's the thing. Like our relationships with our larger neighbors, our relationships with our microbial neighbors are complex, context-dependent, and we still understand them very poorly. We're a long way from being able to categorize microbial neighbors into categories like those you Ways of Knowing students looked at, starting with the parable of the Good Samaritan. Who are the fungal Levites? Or the bacterial priests? Or the viral Samaritans? Who knows? And anyhow, one of the many lessons of that parable is a caution against rigid categories in the first place. As Jonathan Jerry at the McGill University Office for Science and Society wrote a couple of years ago, if you've heard something specific about the microbiome, odds are it's wrong. So today, Rachel and I want to tell you 6 short stories about neighborly relationships between microbiomes and us, their macroscopic hosts. Some of these are feel-good stories, some of them are less so. Some fit the theme of neighbors more directly than others. We won't have time to unpack all of their implications, but hopefully they give us an opportunity to stop and think for a moment. If not now, then maybe later while you're resting. Okay, I feel kind of like Ira Glass, This American Life up here. Story number 1, it takes a village to raise a child. The vast majority of microbes in our microbiomes live in our digestive tracts. particularly in our colons. So you won't be surprised to hear that the microbes living in the digestive system, our gut microbiome, affect the development of the digestive tract itself, or play a role in digesting our food. They do these things. The microbial diversity in our microbiomes is reflected in the fact that our intimate microbial neighborhoods consume and produce a far greater variety of food molecules than our own human cells do. So yes, the gut microbiome certainly plays a substantial role in supplementing our own metabolism and in the development of our digestive system. But consider the ways that your gut connects to the rest of your body. It secretes hormones, it sends signals through nerves to other places, it transports molecules from the lumen, the central tunnel that runs through your esophagus and stomach and your intestines, from the lumen into the circulatory system. And so, presumably, molecules produced by members of the gut microbiome can enter into the general circulation. And microbes living in one part of the body neighborhood can have an effect on other parts of the body neighborhood. Now let's make things more interesting. Because all of us in this room once lived inside another person's body neighborhood, our biological mothers'. In mammals, babies inside the womb take in materials, nutrients, and other molecules by absorbing them across the placenta, which is kind of like a root system growing out from the baby's umbilical cord, bringing the baby's blood vessels right close to, but not quite touching, mom's. Mom, with her gut microbiome, has microbial products that diffused from her gut into her blood, and then from her blood, some of these can diffuse into baby's blood. So given all that, a question that arises is whether mothers' gut microbiomes might influence the development of their children. And in 2020, Dr. Helen Wong and her colleagues at UCLA conducted a set of studies to examine whether the gut microbiome in pregnant mice affected the developing brains of those mothers' pups. Baby mice are called pups. Mice are not people, but they are mammals. Their biology is very similar to ours. So let me give you an overview of what Dr. Wong's research group did. Under standard lab conditions, pregnant mice with typical microbiomes give birth and their pups grow up. And if we follow their pups into adulthood, washed out, but you can imagine what it might show. If we follow their pups into adulthood, those mice will have typical brains and behave in typical ways. In the mammalian brain, a region called the thalamus serves as a hub that collects sensory signals from all over the body and sends them out to the cortex, the outer layer of the brain, which sorts out what to do with all this information. And the thalamus connects to the cortex through a set of nerve cells or neurons that grow out through a region called the internal capsule. A typical mouse will respond to sensory stimulation. If it hears a sudden loud sound, it'll startle. If you put some sticky tape on its foot, it'll get curious about that, and eventually, probably, it will take it off. So, to examine the effect of the microbiome on brain development, Wong and her collaborators worked with 2 additional groups of mouse mothers. In one group, they took these moms and dramatically depleted their microbiomes with large doses of antibiotics to kill off the bacteria living in and on those mice. A second group was raised completely germ-free from birth and never had microbiomes to begin with. So to examine the effect of the microbiome on brain development in the little mouse pups then, sorry, let me just skip over that, and since I already said it. So, if a mother's microbiome affects brain development in her pups, then you would expect that the children of these other groups of mothers with depleted or absent microbiomes would maybe have altered brain structures or behave differently. And indeed, that was the case. In both groups of mothers with the depleted or absent microbiomes, adult children, so we followed these little pups up to adulthood, and then gave them various behavioral tests to take. And they were less startled by loud noises. They took longer to inspect their paws if they put tape on them and to remove the tape that was on them. And they were less responsive to some stimuli like getting poked in their hind paws. Now, other behaviors, like exploring novel textures, responding to heat, or the ability to stay upright on the mouse version of like a rolling log in water, those were not different between these groups. And later on, in autopsies of these mice, the mice from moms with depleted or absent microbiomes had a thinner internal capsule connecting the thalamus and the cortex, and that is sort of anatomically consistent with what you would expect to see in mice with altered responses to sensory stimuli. This study and others since suggest that yes, bacteria growing in mom's digestive system influence the development of her children. But let's be clear, these effects are nuanced. And keep in mind that detectable differences in behavior are not necessarily problematic differences. Also, to see these differences, researchers had to completely or nearly completely eliminate a mouse mother's microbiome for the duration of her pregnancy, starting before the pregnancy and continuing through it. So I just want to underline, in case Donald Trump is listening, that the take-home message here is not that taking antibiotics to treat an infection for a couple of weeks during pregnancy will mess up your kids' brains. Okay, not. But the point is that what we understand to be typical development does appear to depend to some degree on input from our microbial neighbors. I've used a lot of weasel words there, appear, to some degree, some input, but the effect is real, and it's complicated.
Skip to the punchline, yeah. All right, story number 2. Vitamin K and the imperfection of nature. So, this is a picture of my daughter, Anna, present day. And here she is, just a few hours after birth. Anna was born full term, no complications or concerns, 10 fingers, 10 toes, you know the story, right? Picture of a happy, healthy baby, and she continues to be today. And yet, just a few hours before this photo was taken, she received the first injection of her young life, a vitamin K shot. In order to understand why Anna, like nearly all babies in Canada and many other countries around the world, receive a vitamin K shot in the first 6 hours of life, it's worth going back to the first experiments in chickens, not babies, that revealed the existence and significance of vitamin K. The late 1920s and 30s were a heady time for researchers of nutritional biochemistry. It was during this time that significant discoveries were being made about what was important in the diets of humans and also farm animals. Researchers already knew about the importance of calories and macronutrients, that is, proteins, fats, and carbohydrates, what we sometimes call macros today. But it was at this time that discoveries were made about other essential elements of the diet, such as essential fatty acids that we can't make ourselves, and the vitamins that we're all familiar with today. Vitamin C, various B vitamins, vitamin D. Henrik Dahm, pictured here, was one such researcher interested in understanding cholesterol metabolism. He experimented with chicks feeding some a diet devoid of fats and comparing their outcomes with chicks fed a normal diet. He made some minor discoveries about chickens' ability to synthesize and metabolize cholesterol, which was something of a hot topic at the time. But more interestingly, in some of the chicks fed a fat-free diet, he observed spontaneous severe hemorrhaging. That is uncontrolled bleeding under the skin or in the muscles or organs. And this is a picture from his Nobel lecture, and you can see the dark parts under the wing of the chick show a hemorrhage. Dam determined that there must be something missing in the diet that was preventing blood coagulation in the affected chicks. After much testing of known vitamins, Dam determined that the missing factor must be a previously undescribed fat-soluble vitamin. And in 1935, he proposed a new, what he called, anticoagulation factor, which he named with the letter K for the Danish and German spellings of the word coagulation, which was convenient because vitamin C was already taken. A few years later, an American, Edward Doisy, was able to isolate the active compound and determine its chemical structure, and Dam and Doisy went on to share the 1943 Nobel Prize in Physiology or Medicine. Later research yet, particularly in the 1970s, determined that there are actually multiple forms of vitamin K, depending largely on their source, which may be sourced from the diet, in green vegetables, animal products, fermented foods, or produced by various members of the intestinal microbiome. So what does Dam's discovery of vitamin K from hemorrhaging chicks have to do with my baby receiving a vitamin K shot? Well, it turns out vitamin K deficiency bleeding can happen in humans too. According to data from the Centers for Disease Control and Prevention, absent intervention, incidence of vitamin K deficiency bleeding can be as high as 1 in 60 babies, that's 60. This manifests most often between 2 and 7 days old, but can happen within the first 24 hours and up to 6 months after birth. It can look like bruising or internal bleeding, which can also include bleeding on the brain, which you can imagine would be quite scary. Although being able to coagulate your blood is obviously an important function for any healthy human baby, vitamin K isn't always high enough in newborns to do this effectively, creating a risk for this kind of spontaneous bleeding. Vitamin K passes poorly across the placental barrier, which we just learned about, meaning that developing fetuses don't always complete their development with enough stored, which can be compounded by some medications and conditions of the mother, further limiting vitamin K delivery to the fetus during pregnancy. After birth, vitamin K doesn't always pass well through breast milk, and infants aren't eating other foods yet that might be sources of vitamin K, like the broccoli you see Anna eating here, or animal products, fermented foods. And lastly, but importantly, newborns don't yet have the gut microbes that can help them make up the difference in vitamin K, because they only begin to acquire their intestinal microbiome at birth, and it takes a couple of years for it to mature to full functionality. What did we learn here? Well, this is a story about sometimes fatal vitamin K deficiency in human newborns that highlights just one of the many mysteries about this fascinating vitamin that scientists are still trying to work out.
Story number 3. Good fences make good neighbors. I never met my grandpa Abner Brubacher, my father's father. Abner and my grandma Arminta, there's a couple of classic names for you, here comes another one, were farmers near Punky Doodle's Corners in southern Ontario with a family of 7 children. In December of 1942, when my dad was 5 years old, Grandpa cut his hand badly while sawing firewood on the farm. He got quick medical care, his hand was stitched up and cleaned out, or so it seemed. But bacteria nevertheless got into the wound, probably some streptococci or staphylococci that are normal residents of our skin. And they proliferated and spread into Grandpa's blood, and he eventually developed sepsis that progressed to septic shock. High fever, pain, catastrophic drop in blood pressure, clotting within blood vessels that cuts off the blood supply to vital organs. Abner Brubacher died on December 23rd, 1942, leaving Grandma with 7 kids, aged 1 to 14,
and a farm that was way too much work for one adult. Septic shock, also known as blood poisoning, is the result of a necessary immune response that goes too far. To understand why this happens, we need to think clearly about where our microbial neighbors live. Yeah, on our skin and the linings of our internal body chambers, the respiratory system, the urinary and the reproductive systems, and the gut. And we're used to thinking of those internal chambers as being inside us, but in a way, they're still external surfaces. Here's a schematic view of the human body. If you blew yourself up like a balloon and straightened out all the foldy bits. An analogy. Imagine you walk into a tunnel that goes through a mountain. Yes, you are inside the mountain in a sense, but you're standing on the tunnel floor. You're not actually embedded in the mountain's rock itself. Our microbial neighbors, our microbiome, is adjacent to us in the same way. They are in us, but they're still sitting on a body surface, not embedded within our flesh itself. Their place is on the other side of the fences that are our skin and the linings of all those internal chambers. If microbes cross those boundaries and get into the blood or into other internal organs, they have it made. Blood is a nutrient-rich, comfortably warm soup. With those resources at their disposal, bacteria will get serious about reproducing, which they do simply by growing larger and then splitting into two. And then 2 make 4, and 4 make 8, etc. At a reasonable rate of 3 generations an hour, one bacterium can become a population of about 1,000 in less than 4 hours, and about 1 billion in less than 12. And by 24 hours, if that rate of proliferation actually held up, you would have close to 5,000 tons of bacteria. But of course, that doesn't happen because it would require the complete consumption of a small city's worth of people. But you get the point. We cannot afford to give microbes free access to the true interior of our bodies. So for that reason, our immune systems are finely tuned to recognize and respond to signs of microbes in places where they should not be. For example, the molecules that make up bacterial cell walls, those are too large to just diffuse across skin or across the intestinal wall into our bloodstreams. So if those cell wall components end up in the blood and the immune system sees them there, it knows that's a sign that the bacteria themselves must have gotten in. And when those cell wall fragments turn up in the blood or in other internal organs, the immune system springs into action and races to destroy any would-be neighbors that have crossed into the inner sanctum. And most of the time, that response works nicely. The trespassers are eliminated, to use a euphemism, and life goes on, for us. If there are enough microbes in the wrong place, and worse, if they spread, then the immune response continues to strengthen, and it becomes more difficult to rein in. Septic shock is the result of a response to neighbors in the wrong place that has escalated into a kind of body-wide immune panic, as the measures meant to limit and trap the trespassers spill over to damage our own tissues. Eventually, the immune tide becomes unstoppable and the body is overwhelmed. The antibiotic, penicillin, kills bacteria without harming our own cells. It probably would have saved my grandpa's life, but it was not widely available until after the Second World War. In Canada today, even after the development of many effective antibiotics, septic shock is still the 12th leading cause of death, accounting for about 1 in every 20 fatalities overall. It's a stark reminder that although we are interdependent with our microbial neighbors, healthy relationships with those neighbors involve fiercely defended boundaries. It's not that those neighbors are looking to go out of their way to hurt us most of the time, but neither do they really exist in order to help us. And we don't really exist in order to help them. They go about their lives, and sometimes they stumble into opportunities to explore new territory, usually with catastrophic results for them, and sometimes for us also.
Story number 4, strength in diversity. We've just heard from John about what happens when harmless microbes in our body get into places where they shouldn't be. Now I want to talk about what happens when they stay where they ought to, but the microbial communities around them change. John has already mentioned the antibiotic penicillin. I don't think we can overstate just how great an advancement in medicine the discovery of antibiotics was. It is considered by many historians of medicine to be in the top 2 or 3 medical discoveries of the 20th century. It transformed not only the treatment of infectious diseases, but also medical care in the areas of surgery, obstetrics, oncology, dentistry, and many more. The story of Alexander Fleming's discovery of penicillin on a moldy bacterial petri dish is often relayed as a story of serendipity in science. But Fleming, among many other scientists at the time, was in fact looking for a cure for bacterial infections. It's just that he discovered it somewhere other than in one of his controlled experiments. Bacterial infections were a serious and often deadly problem, and medical researchers were eager to identify chemical agents that could cure them. Therefore, it was no surprise that once penicillin could be purified and produced in large enough quantities, people were happy to use it. If you think about your own medical history, I would guess that many of you have lost count of the number of times you have been prescribed antibiotics. I certainly have. In fact, I would be surprised if there was anyone in this room here today who had never had at least one course of antibiotics. And I'm sure you were prescribed antibiotics for good and rational reasons. I'm not arguing against their use. Again, antibiotics were a game changer for modern medicine, and not just penicillin. After its development, many other antibiotics soon followed. The result today is that over 50 types of antibiotics are listed on the World Health Organization's model list of essential medicines. That's medicines that all countries with functional health systems should have on hand at all times. At the same time, we are being warned about the overuse of antibiotics. For example, a couple of weeks ago, the WHO released the latest global antibiotic resistance surveillance report, showing that antibiotic resistance continues to rise, with the greatest impacts observed in the most resource-poor regions. However, resistance to antibiotics isn't the only negative outcome of their use. A more insidious downside to antibiotic use is that it kills bacteria somewhat indiscriminately, depleting the microbiome and diminishing its diversity. This leaves opportunistic microbes without competitors, allowing them to expand their populations to the point of becoming a problem. There are quite a few common microbiome member species that are famous for switching from friend to enemy when the conditions are right. We just heard about a couple. Most of them are bacteria, but for a little bit of a change, let's look at the fungus Candida as our example. You are probably familiar with fungi as food. We eat them as mushrooms, but also harness them when we make bread, beer, and a variety of fermented foods. In natural ecosystems, fungi play a vital role in decomposing organic matter and are among the few types of organisms that can break down wood and other tough plant matter. Depending on the species, fungi can exist in a larger multicellular form, like we see in mushrooms, or smaller single-celled forms, which we call yeast. In our bodies, fungi most commonly present as yeast, and this usually includes Candida. However, a really interesting wrinkle with Candida is that under the right circumstances, it will shift from the harmless yeast form into a multicellular form, sending out root-like structures called hyphae. These hyphae penetrate into the host, causing infection. There are a number of conditions under which this can happen, but an important one is depletion of the microbiome through antibiotic overuse. Although there is still a lot that is unknown about the interactions between bacteria and fungi in the microbiome, it seems clear that in a functioning microbiome community, the relationship between its various members is an uneasy one. Both bacteria and fungi appear to produce compounds that limit the growth of the other organisms that they live alongside, essentially keeping each other in check and playing by the rules. When antibiotics wipe out some of these competitors, other players, like Candida, can be allowed to overgrow, and in this case, causing oral thrush, vaginal yeast infections, diaper rashes in infants, and at its most extreme, even sepsis, meningitis, and endocarditis, which is infection of the heart valves. These are some of the very same sorts of ailments that antibiotics were first developed to cure in the first place. The story of Candida warns us about overusing a good thing, showing antibiotics to be a double-edged sword when it comes to controlling infection. Oh, this is me again. All right. You already heard I have something of a love affair with the country of Panama, which began in 2011. I was working alongside a team of nutritionists and agriculture extension workers from the Panamanian Ministry of Health who were carrying out a project to help families increase and diversify their farm production, which they largely depended on for their own sustenance. The purpose of the Ministry of Health's project was to improve households' food security and diets. In my research, I was trying to understand the complex ways that these changes in diet and the inevitable environmental exposures from this project were impacting children's growth. A big part of my work was to measure parasite infections in these children. Parasite infections are also known to impact growth and are recognized as a public health problem in this region and many others, as we'll soon see. As John mentioned in the introduction, we're host to not only microbes, but a diverse set of animals too. This includes nearly 300 species of helminths, which is another fancy word for parasitic worms. Some of the most common of these helminths falls into the category of soil-transmitted helminths. This was the group of parasites that I was primarily finding in my research in Panama. Worldwide, this group infects an estimated 1.5 to 2 billion people. You can see on this map, though, that the distribution is not uniform across the world. We find them primarily in the global south in areas of lower resources. This group of intestinal worms isn't a single species, but it includes a few different ones: the human roundworm, whipworm, and the hookworms, pictured here. For simplicity, let's focus on the hookworms. They're thought to be the most common of the soil-transmitted helminths, infecting an estimated 500 million people, so that's 500 million people worldwide. Although quite small, hookworms can dramatically impact their host because they feed on host's blood. In the bottom left picture, you can see the head end of a hookworm with its teeth, which we call cutting plates. It uses these to slice into the intestinal wall of its host to access the host's blood. These worms can cause anemia and malnutrition, and these are particularly devastating in childhood and pregnancy, leading to growth stunting and cognitive delays in children, and low birth weight and other complications during pregnancy. Although hookworm infections rarely cause death, the suffering and sickness can be significant. And it can be felt financially, too. Hookworm infections have been estimated to lead to a global loss in GDP of as high as $139 billion annually. As adults, hookworms live mostly in the intestines of their hosts, but as the name of this group, the soil-transmitted helminths, suggests, they have an important developmental stage in the soil. Hookworm eggs are passed out of infected hosts with the host's feces, and in areas with limited sanitation, fecal matter can end up contaminating soil around the home and in farming areas, and contaminating the soil with hookworm eggs. The eggs hatch, releasing larval hookworms into the soil, and over a couple of weeks, those larvae develop into the infective stage. People can become infected when they come into contact with them while walking barefoot or handling contaminated soil. With this kind of transmission cycle, you can imagine why young children would be particularly susceptible to infection. They put everything in their mouths. That's an excellent way to learn about the world, but it's also a really good way to pick up hookworms. Okay, so what could possibly be the upside of hookworm infections? In order to understand any possible silver lining, we need to go back to 1989 and research by the British epidemiologist David Strachan into what he named the hygiene hypothesis. He and other researchers at the time recognized an increase in autoimmune conditions like allergies and asthma in high-income parts of the world. They also noticed that this increase seemed to be less pronounced in rural and farming communities. Others noticed that autoimmune diseases tend to be increasing in prevalence in high-income countries, but not low-income countries. Together, these observations suggested that overall microbial exposure in less hygienic environments could be responsible for the lower incidences of autoimmune diseases, hence the name hygiene hypothesis. In the decade following Strachan's publication of the hygiene hypothesis, researchers were busy uncovering many of the mysteries of the human immune system. Not surprisingly, researchers working on helminth infections also became interested in Strachan's hypothesis. Similar patterns were being seen between helminth infections and allergies and other autoimmune diseases like Crohn's disease and ulcerative colitis. We see in this figure that higher income countries tend to have higher prevalence of food allergies. This is data published in 2020. And if we look at that food allergy next to the helminth infection map we saw earlier, which is from 2023 from the WHO, we can see a rough pattern emerging. I'll let you look at it for just a moment. The darker the red or the brown, the higher the prevalence. So in that top left, we see darker colors in the global south, in parts of sub-Saharan Africa, South and Southeast Asia. And on the bottom right, we see pediatric food allergy prevalence, particularly in North America, Europe. We see some in China and a few other places. Of course we know correlation does not equal causation. This is not evidence, right? It's just a hypothesis. But important research into the functioning and malfunctioning of the immune system over the last several decades has shone light onto the various mechanisms that could be underlying this pattern. We won't go into all of these mechanisms today because honestly, they're really complicated and they're a very hot area of research right now. But we can see in this figure a simplified story of just one piece of the puzzle. We have a representation of a modern, very hygienic sanitation system on the left and of a more rudimentary system on the right. And we see that the system on the left leads to lower exposure to pathogens, but a weaker immune regulatory network, leading to inflammatory molecules and allergic responses and asthma. Whereas on the right, those infections help to train the immune system. So children who were measured showed that they'd been exposed to allergens, but they didn't have the allergic reaction to them. An explanation for the relationship between lower exposure to infections and higher rates of autoimmune diseases is the old friends hypothesis. This is the idea that our species has co-evolved with the microbes and parasites that inhabit us, and our immune systems are adapted to their presence. Without that chronic exposure, Our immune systems lack the training to recognize true danger when it invades our bodies. The long historical relationship between our species and the microbes and parasites that we evolved with has wired us such that their absence can have unexpected and unpleasant consequences for us.
Story number 6. Invaders we can't do without. Viruses are also part of the microbiome, but arguably one we know even less well than the others. Maybe in part because most of the viruses we find in our microbiomes actually infect our microbial neighbors rather than human cells. But maybe also because viruses are not quite living things, and studying them can be tricky. A virus is a little bit of genetic code inside a protein coat, like an M&M. It replicates by getting into a suitable host cell. For example, the COVID virus gets into cells lining the respiratory tract. And once it's in there, the genes that the virus carries are sort of emptied out, and they're decoded by the host cell and persuade that cell to start making more copies of the virus and releasing them, and those can then go on and spread to other host cells and so on and so forth. Often, the infected host cell dies, either because of the stress and damage caused by its new role as a virus-producing factory, or because the immune system recognizes the infected cell as a problem and destroys it. Of all viruses, in my opinion, the creepiest ones are retroviruses, a family whose best known member is HIV, which causes AIDS. I consider retroviruses especially creepy because, unlike other viruses, when one of them enters a host cell, the genes that it carries don't just sort of sit there and get read, but they actually integrate themselves or insert themselves into a chromosome of that host cell. And so the viral genes become part of the host cell's own genetic material. Viruses are quite specific about which kinds of cells they infect, fortunately. For example, HIV infects a specific class of white blood cells called helper T cells, which are central players in our immune systems. So, HIV integrates its genes into and exploits the very cells that would normally protect us against them. Without treatment, HIV can effectively eliminate helper T cells in a person's body, leaving those people with severely impaired immune systems, vulnerable to being overrun by microbes or tumors that would normally be harmless. And so we call the illness caused by HIV AIDS, the acquired immune deficiency syndrome. And HIV is just one example of a retrovirus, one that happens to infect helper T cells. A little thought experiment. Imagine what would happen if a retrovirus manages to infect an egg cell or a sperm cell. At fertilization, if the egg or the sperm cell is infected, then the retrovirus that it carries as part of its genetic material will become part of that fertilized egg's own genome. And since all cells in a person's body arise from a single fertilized egg, all of that person's cells will carry that virus. And we call viruses, retroviruses in this state, endogenous, like internal retroviruses. The good news is that retroviruses rarely infect sperm or eggs. And furthermore, organisms like us do have ways to suppress the activity of endogenous retroviruses. So in at least some cases, we can tolerate having their alien genes in our own genetic makeup. And over evolutionary time, millions of years, lineages of organisms accumulate more and more endogenous retroviruses in a sort of domesticated state. The current estimate is that about 8% of a typical human genome consists of these accumulated creeps. These semi-tamed endogenous retroviruses can still cause problems, Sometimes they reactivate, sometimes they insert into a bad place in the genome. And so research suggests that there are connections between endogenous retroviruses and at least a few different types of chronic diseases. But for the most part, we tolerate them. But in biology, what starts off as tolerance can lead to dependence. And there are also examples of endogenous retroviruses that have come to play essential roles in the lives of their hosts. Of these, and when I first heard this story about 8 years ago, I just about fell off my chair. But of these, the best documented case is the evolution of the placenta in mammals. Of mammals, only platypuses and spiny anteaters, or echidnas, lay eggs. That's not very many species of mammals. For the rest of us, we grow up in mom's womb, as I've previously said, for at least some amount of time, and the developing infant is nourished via a placenta. In marsupials like kangaroos and possums, the time spent in the womb is shorter, but it's still essential. Now, if you take a look at the genomes of mammals, and we've done this now with many, many different types of mammals, one thing that's curious is that all live-bearing mammals, not the platypuses or the echidnas, but all live-bearing mammals share a gene called PEG10. PEG, P-E-G, stands for paternally expressed gene. You get one copy of this gene from your mom and one copy from your dad, but only your dad's copy is active, so paternally expressed gene. The weird thing about PEG10 is that its structure and its behavior are very, very obviously retroviral. It is clear that PEG10 is a domesticated endogenous retrovirus, and we cannot survive without it. In mammals where PEG10 is somehow inactivated or just absent, or if we take it out of some mammals, which we can do, the placenta fails to develop and the embryo dies. So PEG10 plays some sort of essential role in the development of the placenta in the first place. And additional studies of genes involved in placental development have shown that a few other essential genes involved in that process also came from other endogenous retroviruses. Of these, some are found in particular groups of mammals, like primates might have their own, or rodents have their own, and dogs and cats have their own. These group-specific genes seem to have come from different retroviruses that infected those lineages at different points. And they play roles in shaping anatomical features that are characteristic of placentas in different mammalian orders. So the placenta of a cow does not look like the placenta of a human, does not look like the placenta of a mouse. And we think that some of these anatomical differences are shaped by these like group-specific endogenous retroviruses. The consensus is that endogenous retroviruses, invaders that we can't do without, were essential for the evolution of the placenta in the first place and for its later specialization in particular lineages of mammals.
I think we've learned a lot this morning. I hope we've learned a lot this morning. We've heard stories of microbes that we need, microbes that should be harmless but can become a problem, pathogens that should be a problem but nonetheless have some kind of upside. I wanted to highlight just one lesson from each of these stories that gives us an idea about how biologists know, the biological way of knowing. Story one, we heard that you're not wrong to think of yourself as an individual, but we're all dependent on the organisms that make up our microbiome. And this starts even before we are born. Story two, we saw that nature isn't perfect. We sometimes hear about the, quote, wisdom of nature. But in fact, what we experience as natural was simply the most adaptive version of whatever options were available at the time that thing evolved. And it doesn't necessarily line up with what we would wish for or design ourselves, such as vitamin K deficiency in newborns. Stories 3, 4, and 5 showed us that whether something harms us or helps us often depends on context. where something is in the body, in the case of sepsis, what else is around in the case of candidiasis, and how long our species has been living and co-evolving with something, as in the case of our old friends, the hookworms. And finally, in story 6, we saw that sometimes things that can seem really, really bad can lead to some surprisingly good outcomes. We often want biology and science more generally to give us clear and simple answers to important questions. What we see in all these stories is that's not how science works. The scientific method, which biological research employs, is always working to refine our understanding of the world, introduce nuance, by rejecting hypotheses that are obviously wrong and continuously questioning what we think we know to come to a better and truer understanding. This means science in action can be disorienting. Maybe you remember this from the early days of the COVID-19 pandemic. There were months in that first year when it seemed like we'd all get whiplash from trying to follow the public health recommendations. Don't mask, mask. Use this kind of mask. No, this other kind. Schools should be closed. Schools should be opened. It was wild. Eventually, enough was known about how this new pathogen behaved that researchers could more confidently advise the public, and technologies like vaccines could be developed. But even after the dust had settled and life returned to some semblance of normalcy, there was still the other issue of science for society to contend with. That the way of knowing, that is the biological sciences, cannot answer some of our more important questions, because they simply aren't scientific questions at all. Should vaccines for COVID-19 have been mandated, or was the cost of individual freedom of medical choice too high? Is the minute risk from vitamin K injection worth taking to protect a newborn from another bad outcome, spontaneous hemorrhaging, which is also unlikely to occur? What is a bad microbe? What is a good microbe? And are those labels even helpful in the complex, multi-species world and bodies we inhabit? These are questions we can only answer together in community with our human neighbors.
Thank you, John and Rachel. My head is very full. Perhaps your heads are also very full, but we have a few moments for a few questions. There are people with microphones wandering around who can either let you use the microphone or repeat your question for you. So a few moments for questions. Does anyone have a question for John and or Rachel?
Do you have favorite microbes?
A favorite microbe?
And what does the microbe do?
Another story.
Do you want to answer? You're the parasitologist.
Not the microbiologist.
So I guess the cop-out answer is I love all microbes equally. Like my children, or my students. I have no children. I mean, it depends on the day. Some days, the influenza virus is my favorite, but that's only in a very intellectual kind of sense because it's a real jerk most of the time. I'd probably have to put a plug in for E. coli as well, because I work with those little bacteria a lot in the lab, and they're kind of endearing in their way.
I don't have one.
We have a question up here, Delmer.
Thanks, guys. I was wondering, did the virus pop in first, or did it pop out first, chicken or the egg?
So, thanks, Travis. That's it's a nice, simple question. So that's a hot topic. I think you can get virologists into some big fights over that one. I'm not exactly a virologist, so I'll just wade in. I think that viruses came later. That viruses are sort of like split off, decayed, partial versions of host cells. So then what? Yeah. No, I don't think so. Nope. I'm just going to say no. There you go. There's a simple answer. We'll talk later.
Yeah, I've got one about, you mentioned it briefly, John, about, there's things that the people are trying to sell us, like probiotic, like probiotic yogurts, big for the last decade, kombucha, good for gut health and stuff like that. And so it's definitely a commercial thing that they're using. But you had mentioned, I want to just expand a little bit, that they're not really necessarily working anyways because the microbes are so complex, kind of thing. So it's obviously just like anything, like the green revolution. They're just trying to sell us stuff by saying everything's green. So can you come out on the record and say all this probiotic stuff is nonsense, or do you believe in it?
Rachel, do you want to answer? On my behalf?
Why don't you go first and then I'll repeat it.
Okay, yeah, good. I mean, it really is complicated, right? My hunch, and I think what research would support is that, like in most cases where you have a well-established microbiome in your gut, you take in these probiotics and you shit them right out. So that's kind of my take, but they're not bad for you, and I mean, who knows? Who knows? But I don't know. It's like, is peanut butter good or bad? Like, yeah. Rachel, do you wanna refute me?
Yeah.
Good.
No, I'll just, yes, it's complicated. It's very complicated, Craig. I will just say a lot of those probiotics have lactobacillus in them, which is one of the first things that inhabits your body. And you have a lot of it, and it does a good job of keeping other things in check. So it's not bad to have more lactobacillus. It's not like you're taking a probiotic of C. difficile or something, right? You don't want just any microbe in your probiotic milkshake. But the other thing about fermented foods is that, like with vitamin K, bacteria are shedding or excreting all sorts of metabolites of different sorts. That we don't necessarily make ourselves or in very high quantities. And so we're not just getting the bacteria there, we're also getting, other kinds of micronutrients, possibly. But yeah, no, the wish for it to be a magic cure is one that we have always lived with. And it's no more true for probiotics than it was for, I don't know, arsenic or whatever people were into 200 years ago.
We have time for one more quick question.
How about up here?
Hello, I'd like to just follow up about that. What if, about the whole kombucha thing, what if you have just finished a round of antibiotics?
I think there's evidence for it, but it's not a perfect fix, right? And in addition to that, you're probably better off focusing on getting a diet that is high in fiber and materials, which is what most of us are missing and what our microbes really prefer to be eating. So we're probably better off thinking about our whole diet rather than trying to pinpoint that one easy thing that we can just buy, right? It's a lot easier to buy that one fancy drink in the grocery store than it is to cook and grocery shop and make things that aren't cheeseburgers but are better for our gut microbiomes.
One other, just to piggyback on one of Rachel's points, which was the importance of having a diverse microbiome. The probiotic, if you take that after you've had antibiotics, you're most likely introducing a whole bunch of like reasonably friendly bacteria into your digestive system, and that's great. But it's going to be a pretty, not quite a monoculture, but maybe just a few different kinds of bacteria. It's not going to give you the full diversity of bacteria that you would eventually get longer term. But in the short term, I say go for it. There we go. I'll put myself on the record there.
Thank you, everyone, for being here. People have to get to classes. Thank you for being here. And a big, big thank you to Rachel Krause and John Brubacher.
Bravo.